So thank you everyone for rushing back from your 1-hour lunch to come hear my talk. And before I start to get into it too much, I just want to say that this is preliminary data. I just got it last week. This is ongoing research that I'm doing for my PhD dissertation. And interestingly, what I found was different than what I was expecting. So yeah, got a lot more questions to answer. So I'm talking about Baetis tricaudatis. And so to just kind of get you guys centered on where we're at, we've been talking about fish, we've been learning about different agencies. So Baetis is a mayfly within the order Ephemeroptera. In the family Baetidae, they're known as small minnow mayflies. They're pretty short-lived as adults, anywhere from like 24 hours to a couple days. I had a couple hang out for 3 or 4 days, and I was raising them on my own. They're really large and widespread genera, so they occur all across the world except for in New Zealand and in Antarctica. And they mostly occur in lotic habitats, but they can be in some pools as well. And this group in particular is interesting because there's a lot of taxonomic problems. There's been a lot of revisionary work that's been going on pretty much since the genus was first kind of discovered. And now with genetics, it's just been really, really hectic. So just to kind of drive this point home, I wanted to highlight some of the many papers that have worked on revisions within this genus. And so my work is kind of another stone in the wall, so to speak. But talking about conservation and biodiversity the last couple of days, I've really been able to get some context for my research and thinking about, why have I been spending years doing this? Why am I putting my life energy into this tiny little mayfly that, aside from a pretty nerdy group of people, a lot of people don't even know about. But I think it's really important to understand, what this species really is. So the species I'm talking about is Baetis tricaudatis within this genus. And they really have a key role in ecosystems, both terrestrial and aquatic. So when they're larvae, we kind of study them. Obviously, we're all working on fresh water here. They're important grazers of periphyton, and they're prey for fish. And of course, when they emerge, they're important prey for birds and bats as well. They're widely distributed across North America. A lot of the work has been actually on the East Coast. And then there's been some work with this species in Southern California. However, in Northern California, there's been very little work done on it, taxonomically or genetically. And so I've been kind of filling in that hole a little bit with my research. One of the reasons, there's a lot of reasons that I think this particular taxa is really interesting. It's important in ecosystems, it's really common in bioassessment and biomonitoring programs, and we can't even identify it down to species when they're larvae, except maybe sometimes if it's like the last instar, and you can have the gills mounted onto a slide, and it can be really specific, but there's been a lot of issues with identifying these. And so I know these are adults, and I've been working with larvae, but these are images from Jeffrey Webb, and hopefully the colors are coming up that you can see that there's a lot of morphological differentiation for the species. It's one of the reasons why it's really difficult to identify. And again, a lot of the work has been done on adults on the East Coast. And so here in California, when we're working on bioassessment and biomonitoring programs, we're trying to do identifications that are consistent, that are reliable, that can be controlled for quality, and that we can really trust. And so that's kind of where genetics really steps in. And I get really excited about these new tools that we can use to better protect and understand our ecosystems. And for me, DNA barcoding was one of those things. And so DNA barcoding, I'm not sure, how many of you guys know about DNA barcoding? Okay, so maybe 20%. Okay, so the CO1 gene is the barcoding gene, and it's a mitochondrial gene segment that was heralded as the universal way to identify all species in a paper by Hebert et al. In 2005. And then there was kind of this big hurrah about, oh, well, the taxonomists don't agree with this, and you're really taking out like a lot of the human element to that. And so I kind of wanted to tease that apart a little bit. And I was just talking to someone as we were moving in here, and there's a lot of pressure sometimes to go into like the newest and greatest kind of scientific find. And sometimes you're really missing what these techniques are trying to actually do. And so I really wanted to tie together genetics and morphology and life history with my sampling and really be able to find out about the diversity of these species. There's been some unpublished work done by Jeffrey Webb, about maybe there being cryptic species within this single species. And that would be really interesting. So that brings me to my study questions. So the first study question is, what is the genetic diversity of the CO1 barcoding gene segment for Baetis tricaudatis within the Russian River watershed? Next, what is the genetic diversity between Baetis tricaudatis and closely related species that maybe we can't tell a difference apart morphologically? What do the genetics tell us? And how is diversity arranged temporally? So this is kind of the meat and potatoes of this presentation, and again, this is just part of this project. But in order for us to really be able to use genetics for our use and conservation, we need to understand it both spatially and temporally, how the genetics change with time, what it looks like for possibly a cryptic species that's really diverse. And then I also, to a smaller degree, looked at how it's arranged spatially. And then kind of looking at this, is there evidence to support cryptic diversity within this population? And so my study location was in the Russian River watershed, which I'm sure many of you guys know about, but it's west of here. It's really close to where I'm from. I'm from Petaluma, and it's really beautiful, and it's a pretty well-managed watershed, too. There is a dam on the main stem, and there have been pretty healthy trout populations. So here are my 3 sampling steps. Oops, I did not mean to do that. No. Giving you guys all the answers to my questions already. Okay. So maybe I won't use that one. My 3 sampling sites were within Salmon Creek, which is right near the city of Bodega. It's pretty coastal. And then Austin Creek near the city of Cazadero, and then the Russian River on the main stem. The first 2 sampling sites are both tributaries of the Russian River, and they're characteristic of the Mediterranean ecosystem that this area is known for. So the first step is the fun part, going out to collect. This is actually my dad. He goes out with me every time. And so over here on the right, we have Salmon Creek. And then in the middle, we have Austin Creek, which is one of the riffles that I sample. And then here we have the Russian River at Sunset Park. And Salmon Creek over to the right, I've been collecting over 3 years. I have 38 sampling trips now. And I haven't been collecting consistent Baetis populations from Salmon Creek. So for the most part, it's kind of on the background compared to Austin Creek and the Russian River. And I have 2 more extensive sampling trips within all of these, and that was really in order for me to get a lot of Baetis tricaudatis so I could capture as best as possible all of the genetic variation within those populations. So I have about 50 sequences from 2 different site times. And so after I get to the fun part of collecting, then you actually have to get into the lab and generate your sequences. And this is something that some people can send off, but I've been really generating my sequences on my own. And so there's been some positives and negatives associated with that. Of course, it's science, so sometimes your advisor's like, so I haven't heard from you in a couple of weeks, so where are all the sequences? And I'm like, well, none of them have worked, unfortunately. So it's interesting because, sometimes the genetics, it seems like, oh, this will be really easy. You just throw it into a next-gen sequencer, and then you'll have the answer ta-da. But I found that, for me at least, it has been a little bit more challenging than maybe it seems like it could be. But I store everything in 95% alcohol. I morphologically identify them. I've been using the universal primers for CO1, LCO, and HCO. And then I've been using standard molecular techniques to go through all of the different steps. And I'd be happy to talk about those with anybody. There's a lot of kind of gray areas in terms of what the annealing temperatures are and what types of products to use, so I'm happy to go into that later. But I ended up collecting 469 sequences across 37 months. I analyzed them in a program called Genius, and I edited them there. And then I would blast them, which is basically just comparing the sequences that I got to other sequences uploaded online through GenBank. And then I did my genetic analyses using Arlequin for the most part, and then PopArt. And I used PopArt to create haplotype networks, which I'll be showing you guys today. So for the first question, the results, this is the exciting part. So what is the genetic diversity within the CO1 gene segment? And with the idea that maybe this is a cryptic species, maybe this can be super diverse, and I found that it was 2.3%, which is exactly what you would expect for a single species at this gene segment. And So interestingly, this 2.3% only occurred at one site and 1 month apart. So I was kind of thinking that maybe between watersheds I would see more genetic divergence, but that wasn't the case. These seem to be a pretty well-mixing population. All right, so what about genetic divergence between Baetis tricaudatis and then closely related species that morphologically show very little differentiation sometimes? For Baetis tricaudatis to Baetis odonis, which is its sister species, which is one of the species that, again, you can't tell the two apart. I sometimes get the two confused, and I've been looking at these things for years, only these things too. And so thinking about for biomonitoring programs, people are looking at thousands of different taxa from different locations. This can be a real interesting thing. So, but the genetics show that they're definitely different species. 10% is a pretty good value for different species. But then you see within the same genus, Baetis, Baetis tricaudatis to intercalaris, you're having over 20%. So maybe that's showing that potentially there needs to be some more taxonomic and systematics work done here, because this doesn't seem right. However, between Baetis and different genera looks about right. And so now to the fun part. And I don't know, these aren't colorful. The colorful ones are coming next. But these are haplotype networks. Now, the size of the circles represent how many individuals are within that sample. And then the dash marks, the length of the lines don't matter, but the dash marks, those are the number of base pair changes. So you can see that for fall 2004, there seems to be more genetic diversity than spring. And now what about looking at this, looking at all of the different population, or all of the individuals that I have, and I was expecting to see maybe by year, so we have 2012, which is just a little bit of the red slice. I started sampling pretty late toward the end of that year, so I have a small sampling size. But again, the two main haplotypes are represented in all of the years, and it's a pretty even proportion, which is really what you'd expect for a well-mixing population. And so I thought, okay, well, maybe year to year, this is what you see. What about different months, or different seasons, or different cohorts? And again, the two main haplotypes represented are differentiated by two base pairs, and then pretty good mixing. So season 1 is basically representative of the first cohort. So from December to March. Season 2 is April to July. Season 3, which is the purple, is August through November. And again, lots of mixing here. So all of these together, next is what about how they arrange spatially? So again, temporally seems to be good mixing. Between years, between seasons. What about spatially? And so I was, again, kind of expecting to see some more kind of structuring associated with the haplotypes. But it looks very similar. And again, we have the two main sites, the Russian River, which is the dominant kind of yellowish green, kind of ugly color, actually. And then the Austin Creek. And this is a little bit misrepresented because of the number of individuals from each of these sites, but the two main haplogroups, again, are present in different watersheds and different areas in Northern California. So again, not what I was expecting, but very interesting. So is this in support of any kind of cryptic diversity within Baetis tricaudatis populations? No, it's really not. It seems to be that these little guys, you can have tiny little centimeter-long wings and who can barely live for a day are actually mixing really well between watersheds. And so, for every question that you have answered, again, this is preliminary results, there's a million more ways that you can kind of look at this data and a million more questions to answer. So in particular, I really want to dive into the spatial patterns a little bit more and comparing northern, southern California, west versus east coast, and then really tying the kind of haplotypes that we're seeing and the diversity that we're seeing, tying that to habitat availability and quality, like flow and temperature. I've been finding that the distributional information that we have doesn't line up with the genetics that I'm seeing, so that's kind of another whole can of worms I want to dive into at some point. And then BLAST, the way that the genetic sequences are uploaded online, there isn't very much quality assurance that goes along with that, and so it can make the data set pretty difficult to rely on. And then also looking at the proportions of similar species through time. And with that, I would like to thank my advisors, and my lab mate Mike Peterson has been doing similar work, so we've been in the lab late many times together, and I've appreciated his support, and also my dad, who's been sampling with me for the last 3+ years, and thank you for your attention. time for questions? Or?
Keep moving.
Okay.
Thanks very much,
Thank you.
Natalie.